Hello everyone, welcome back to the channel. So today we'll be discussing a few important points from the topic genetics and health for community medicine. So a few cytologic facts. Tijo and Levin reported that our genome has 46 chromosomes, which can be divided into 7 groups A, B, C, D, E, F, G. So group A has 1 to 3, B has chromosomes 4 to 5, C has 6 to 12 with the X chromosome, D is 13 to 15, E is 16 to 18, F is 19 to 20, and G is 21, 22, and the Y chromosome. Next, we should know what is mitosis, meiosis, and chromosomes. We also know about the centromere, pairing, crossover, haploidy, and the methods of chromosomal study. We then see genes, the loci, the concept of homo and heterozygosity, dominance, recessive, polygenes, and penetrance. Then we see what's a genotype, a phenotype, what are chromosomal anomalies. Due to non-disjunction, we can have trisomy or monosomy. We can also have translocation, deletion, duplication, inversion, and isochromosomes, and mosaicism. Coming to the laws of inheritance, we have the law of inheritance of unit characters, the law of dominance, and the law of segregation. Then we have genetic disorders, which we study in detail. So chromosomal anomalies, we have disorders of the sex chromosomes. Two classics come to mind, that's Klinefelter's and Turner's syndromes. Klinefelter's syndrome deals with 44 autosomes and an XY genotype with extra X chromosomes. In Turner's syndrome, on the other hand, you just get one X chromosome along with the 44 autosomes. If you get an extra Y chromosome, you're a super male. If you just have extra X chromosomes with an XX genotype, it becomes a super female. Coming to autosomal anomalies, we have trisomy 21, that's Down syndrome, trisomy 18, that's Edwards syndrome, and trisomy 13, that's Patau syndrome. Down syndrome is also known as Mongolism. And Mendelian diseases, we have the 3 Mendelian laws. We deal with point mutation. There's a small table of autosomal, allosomal, dominant, and recessive diseases. If you read this table, you are sorted. We then discuss the ABO system, rhesus system, and various other blood groups and how they're related with disease, and erythroblastosis fetalis and other diseases like sickle cell anemia, thalassemia, hemophilia, cystic fibrosis, and PKU or phenylketonuria. We then see the multifactorial causes of genetic disorders and genetic predisposition in common disorders like cardiovascular disease, etc. And then we see the application and uses of genomics like gene therapy, the Human Genome Project, and the Human Genome Diversity Project. There's a small table for this also. And then factors influencing gene frequencies, we have mutation, natural selection, population movements, breeding structures, and public health measures. We then move on to the preventive and social measures relating to genetics. So in health promotion, we have eugenics, which can be negative or positive. Hitler's Jewish genocide is a good example for negative eugenics. We then also see what's euthenics, genetic counseling, which can be prospective or retrospective, and other measures like avoiding consanguineous marriages and avoiding late marriages as well. And then specific protection, we also have early diagnosis and treatment like detecting genetic carriers, prenatal diagnostic techniques, and the indications for which is the table, and a small note on amniocentesis, the screening of newborn infants, and recognizing preclinical cases, and of course rehabilitation and disability limitation. There's a small table for this as well. So that brings us to the end of this chapter. Hope this is helpful. Thanks for watching, guys. See you soon.